Hi, everybody. My name is Tama Hassan, and I am the Assistant Dean for Undergraduate Research here at UCLA within the Division of Undergraduate Education. I'm also the Director of the Undergraduate Research Center for the Sciences, and it is my pleasure to welcome you to our final special keynote speech that's part of our third virtual Undergraduate Research Week. Now that we're at the end of the week, I can share with you that we have had over 5,000 views of the various videos that have been produced by our undergraduates showcasing their research. And you can still watch those videos if you go back to the showcase, click and search through all the different disciplines. And on Tuesday, as part of our research showcase, we had many thousands of visitors attend the 110 panels where our students shared their work orally. Again, in every discipline under the sun. So it's been really great to hear from our alumni and our faculty and all the different friends and relatives of our students as they've shared their different perspectives. But today, I have the pleasure of introducing a student that I mentored as an undergraduate, who's going to be coming here as an assistant professor in a couple of months. His name is Dewan Farmer. And Dewan is a developmental biologist. He will be joining the Department of Molecular Cell and Developmental Biology in July. Now, Dewan majored in MCDB, Molecular Cell and Developmental Biology, when he was an undergraduate here with a minor in biomedical research. He was one of the very first of our biomed research minor students. He was a member of MARC, Maximizing Access to Research Career, and he worked in a research lab doing developmental biology, as you'll hear, on vascular biology. He went on to get his PhD in biochemistry and molecular biology from UCSF, and he completed his postdoctoral studies across the other side of LA at the University of Southern California. But he's coming back to us, we're thrilled to say, and I would like to have all of you welcome with me to join Dr. Dewan Farmer for his presentation. So Dewan, please unmute yourself and we're ready to hear your story. We're so happy to have you with us today. All right, I'm going to stop my video so they can see your screen.
Okay. Well, thank you so much, Tama, for that really warm introduction. Hello, everyone. I'm very excited to be here with you all. Hopefully you all can see my screen. And so, yes, I'm excited to be able to participate in Undergrad Research Week, which is where I started doing my first presentations in scientific research. And actually, I'm officially here and in my office at UCLA. So I'm super excited to be here with you all now. And so today I'm going to take you all through the journey for me to actually discover scientific research as a pathway for a career. And I'm going to show you how I started and how I allowed the process of scientific discovery to really dictate what I did for my career. And so I need to kind of set some context because about who I am, because that really shaped my introduction into scientific research. And so I am a first generation student from Compton, California. I'm an African-American male, and these were part of my salient identities that really defined my entry into UCLA in 2006. But other than these markers, I also, at the time, was very much interested in medicine. I wanted to be a physician. I really wanted to be able to actually treat patients and work with patient care. And within that spectrum, I remain very interested in advocacy and social justice issues. And in particular, I entered with a very pure interest in understanding the origins of health disparities. And so these are the things that I was interested in when I started my work at UCLA. And really at that time when I started, I really didn't know what research was. I didn't really know it existed. I had no idea that the professors here were conducting research. I really came to UCLA because I was pre-med and wanted to be a physician. But, and really a story I don't have time to share with you all in detail, I really stumbled into research. And I met this, this person who really opened up and allowed me to join her research team to begin to ask questions that were related to my interests at the time. And that was Dr. Vicky Mays. And really what I was working with Dr. Mays to evaluate was the actual social influence on health disparities. In particular, trying to understand how perceived discrimination might actually affect health outcomes. And so here I'm just showing you, and so when I'm showing you data, I'm showing you data that actually came from slides that I created and presented at some UCLA presentation as an undergrad. And so this is data that was collected in 2006, 2007. And so even though it's from 2005, it was very, very recent at the time. And so here is a map from the California Health Survey, or a graph really showing that what is now commonly understood, that compared to other races, African Americans have nearly double the chance of having had high blood pressure or hypertension. And we know that there's a lot of known causes for hypertension, like age, obesity, alcohol consumption, diabetes, inactive lifestyle. But one of the well-known causes is also stress. And so what my research and what my pertained to was beginning to understand the origins and how perceived discrimination might be transitioned into a type of stress that can influence health conditions. And here is just an older graph from the Houston area survey that demonstrates that broadly, it is broadly considered, more than by half the population that was surveyed, that Black people in the US have a long way to go to have the same life chances in life as white people do. And so this has a lot to do with the way that people perceive America and how they perceive circumstances. And just an interesting note, this has actually gone up in the past recent years and not gone down. And so what I was actually, oops, I'm sorry, sorry, hopefully I'm still sharing. Sorry about that. And so what we were doing was beginning to understand the mechanisms by which this process of perceived discrimination might be processed into a physiological response. And really the basis of my research was, and this is a lot of reviews and creating surveys to begin to assess a process called allostatic load. And allostasis is actually the process by which you recover from a stress-related experience. And what this graph is kind of trying to demonstrate is that there's a lot of markers or influencers for how you actually respond to a stressor. And so one of these is the external environment, your actual individual differences, like the genes that you have, or the different mutations you have in your genome, but also trauma, abuse, and how you behave. All these influence how you're going to return to your baseline homeostasis in this process of after receiving stress. And allostatic load is when this process is not properly turned back on. And so here, this is a normal condition where there's a stressor, and then you have a response, and this is a physiological response where you have hormones that are being released in response. And so one of the key hormones that we think about in the context of hypertension is elevated cortisol levels, which is something that, of course, that comes from the hypothalamus to the pituitary gland to the adrenal glands to release the cortisol levels. And so here you have a stress, you have a response, and then you return back to baseline. But in the context of allostatic load, you might be actually having repeated hits. And so for a person maybe receiving racial discrimination, maybe you're receiving active repeats of this discrimination. So you're actively consistently on and consistently perceiving stress, which means you have higher on average rates of this cortisol. An alternative is that you have this other like prolonged response. And so now you have experienced it, but now you are having problems returning back to that, that homeostatic condition. And so this was what we were investigating, and particularly what I was beginning to think about was how police Interactions with the police might be, and perceived discrimination in that regard might actually be leading to higher increased allostatic load in African-American men. And so this was my introduction. And this actually really nicely packaged all my interests at that time in a research program. And so this was really, really exciting. But what I found while doing this research is while I was really having a blast and having great peers doing really cool science research, I found that what was really becoming interesting to me was beginning to understand the details at the molecular level of how these defects or how cortisol is actually affecting and causing changes in the vasculature to actually cause things like hypertension. And at the same time, I began to transition, and because I actually participated in a program that was run by Tama Hassan, who introduced me today, called BICEP, and I got hands-on experience doing lab work. And so then I was trying to think about, can I actually transition to a basic science research program to understand if how vessels actually experience the actual signals that are important in perceived stress and perceived discrimination. And so I ended up joining the laboratory of Dr. Louisa Arispe to understand the vasculature and blood vessels. And actually, interestingly enough, I was put on a project that was focused on the placenta. And so the placenta is the interface between the mom and the child and plays a very important role in providing oxygen and nutrients to the embryo. And in addition, throughout pregnancy, we already know that hormones are incredibly important for pregnancy, and many different hormones go up at different stages throughout this process. And what my project ended up being was an intersection between hormone regulation and the placenta vasculature. And so here I'm showing you the labyrinth or the vasculature for the placenta here. And this was the region that I was particularly interested in. And this research actually came from an observation that was made from my postdoc mentor, Anne Sylvain, within Louisa's lab. And so what she did was she was doing an experiment and she added this drug called tamoxifen. And tamoxifen is it interferes with estrogen signaling. It can either activate it or it can antagonize it, depends on the context. But she added tamoxifen at this very specific window, E6.5. So this is after the embryo has implanted, but during the time when like the placenta is really beginning to form and that vasculature is beginning to be established. And when she added tamoxifen at this time point, and she looked a few days later, she found these are just individual embryos wrapped with their placenta and their yolk and the uterine horn. And what you see in the treated animals, you see that between the actual embryos, you see this mass of accumulated blood. So this suggested that there was something wrong with that somehow tamoxifen was interfering with the vasculature. And so for the first time, this is my first experience working with any kind of animal model. And so it was really exciting because we were able to dissect out animals and we would look at the placentas. And particularly on the uterine side, we found that the spiral arteries, which really supply the blood to the placenta, were very clearly abnormal. So normally they have this very clear spiral shape. They call it spiral arteries. And in our treated animals, they clearly look irregular and elongated. So this is a very clear sign that there was something going wrong. And then for the first time, I was able to do experiments like immunofluorescence, look at the protein within a section. And we were able to see less dense vasculature within the treated animals, demonstrating that estrogen signaling is important for properly establishing the proper architecture to support the vasculature in the placenta. And so this was a really, really exciting experience for me. It was my first basic science experience, and I stayed in the lab for a few years. I think I started in 2008, 2009, and I became a Mark Fellow, but I also did programs like SURF in the summer to continue this. And I also participated, like many of you all, in Science Days. And so here's a couple of pictures from Science Day. And this is me maybe a year or so, maybe 2008, 2009. And this is my senior year. And you can see that both the poster evolved And it was really fun because I was really generating all this data on my own. So this is a really, really exciting experience for me. And that's just a throwback for you all to realize where I was in exactly your same shoes, quite a few, over a decade ago now. But what this did for me was it basically demonstrated that I really, really enjoy doing basic science research, and that instead of actually focusing on practicing, I decided that I wanted to actually pursue research as a full-time career and actually really focus on getting my PhD instead of going to medical school. And I wanted to focus on the vasculature. And interestingly enough, I'm going to tell you, but I don't study blood vessels today, but in graduate school, you do rotations. And so you go through different labs and you decide if you want to join that lab and get some experience. And I rotated in all the vascular biology labs that were many of the vascular biology labs at UCSF. And I ended up choosing the one lab that was not a vascular biology lab to join. And so this, again, is allowing, and it's just because the science caught me and I really enjoyed what I was doing in that lab. And so I ended up joining a lab that studied RNA. And in particular, we studied a type of RNA, these small RNAs called microRNAs, which were at the time were thought to be important in regulating biology, and has been proven to be true many times since then. And we were studying this particular microRNA called MIR205. And because when we knocked it out, we found that half the animals looked like this little guy here. The skin was underdeveloped, and these animals are small, would eventually die. And so this was a really cool observation because it showed that this 22 nucleotide microRNA was able to be so important that you wouldn't be able to survive without it. But what I became interested in is the surviving population, because if I looked at these animals and I compared them to controls, these animals' eyes look really wonky to me. And I became really intrigued in figuring out what that was. And what it looked like was that these animals were rubbing their eyes, so that's why they have less hair around their eyes, because they might have something called dry eye. And we all know what dry eye is. It's when you don't have proper lubrication of your eyes. And so we did a test where we tried to induce them to secrete tears. And when we were very surprised to see that compared to controls where they were able to produce lots of tears, the mutant animals never produced any tears. And so this was a big surprise and really kind of transitioned my whole research program to focus on this structure called the lacrimal gland, which is important for secreting tears. And so what we ended up finding was that compared to controls, where you can see this lacrimal gland in green here, these animals never formed a lacrimal gland. And so this was a really exciting development. And what it did for me was it really centered the rest of my time at UCSF on developmental biology. And there, I went on to begin to think about progenitors and stem cells, but I also began to start doing really cool cutting edge techniques like single cell RNA-seq. I began to work with human tissue because we got human lacrimal gland tissue. But most importantly, what this work did was it really demonstrated to me that even though I hadn't seen a pattern between my undergrad research and my graduate research, I began to realize that I was interested in understanding developmental biology and how tissues are established to form the complex forms that support our body's functions, and how this developmental process can go wrong to cause disorders, defects, and disease. And so from that standpoint, I really began to understand that developmental biology is where I want to be. And for many folks who graduate from with a PhD, they often go on to pursue a postdoc. And I decided to do that, and I wanted to do it in a hardcore developmental biology lab. And I wanted to actually use a system that would allow me to look at developmental processes throughout the lifetime of the animal. And so for that reason, I wanted to join a lab that studied this species called zebrafish. So zebrafish are vertebrate animals. They have a skeleton. They have a lot of the same organs that we have as humans. But unlike humans, unlike mice, these animals develop outside the mother. So here we can see that this embryo starts as a single cell like we all do. And in 48 hours, it has gone from this single cell embryo to this multicellular organism that has an eye, that has a heart, that has a brain, and that can begin to move. And so this was a powerful tool for us to think about. And so I began to interview in Zebrafish labs. And again, I applied to a lot of labs that did not focus on, that focus on vasculature. That I wanted to go back to the cardiovascular system because I still wanted my research to actually be able to begin to help me inform health disparities. But I ended up joining the lab that studies the skeleton. And it was because the science in this lab really drew me. And so so that despite the fact that zebrafish look very different from humans and from mice, a lot of the different components of the skeleton, like tendons, bones, and muscles, are also part of the skeleton in a zebrafish. So we can use that core similarity to understand the basis of disease, but also because the zebrafish develops outside the mother, we can watch processes take place. So here I'm showing you an image that was taken from a member in the Crump lab, where you can see the actual cartilages begin to form and take shape. And so this is a really, really cool and exciting development. And so, and I particularly began to study a structure in the craniofacial skeleton called the calvaria. And so the calvaria is this really important structure that protects the head from the external environment. And it's comprised of these 5 different bones: the frontal bones, the parietal bones, and the occipital bone. If we look from the top, we can see that there's these 5 bones, and we can see you have these little wriggly lines that are supposed to represent these structures called cranial sutures. And these sutures will act as boundaries that separate these bones, because these bones need to keep growing over time as the brain grows. And in a craniofacial defect called craniosynostosis, one or more of these sutures can be lost. And as a result, you restrict the growth of the brain, of the skull, while the brain is growing. So you have increased intracranial pressure. And to correct this, surgeons have to go through and break apart these bones so that these patients can actually have space to accommodate the ongoing growth of the brain. And so my postdoctoral work, as well as my own lab, is focused on understanding the underlying biology that is important for establishing these structures, but also beginning to understand what happens when these structures, what drives these structures to be lost in this common congenital defect called craniosynostosis. And so what we use now is we're always interested in using cutting edge technologies to begin to actually understand how these structures called the calvaria form. And now I've combined a lot of the features of my graduate and my postdoctoral work, because I use both mouse and zebrafish models to begin to understand the basis of this human congenital disorder. And again, building from my work as a graduate student, I'm interested in understanding the stem cell behaviors at these in the calvaria, as well as understanding the key signals that are important and how these processes go awry to cause disease. But I'm also actually interested in the blood vessels that support the calvaria. So I'm still going back to my early underpinning interest, but still you can see that there's been a lot of unexpected turns and developments that have really pushed me quite far away from where I thought I would be in 2006. And so now when I compare myself from my 2006 self to my 2022 self as a first-year professor at UCLA, firstly, there's still my salient identities. I'm still first-generation African-American male from Compton, right? But now, instead of focusing on trying to be a physician and studying medicine, my passion and what I want to do is understand developmental biology and what are the systems that properly establish a functioning organ and how that goes awry in human disease. And instead of focusing on social justice in terms of healthcare, I'm far more focused on thinking about equity and inclusion and in higher education. And then finally, instead of thinking about health disparities, I think about disparities in STEM education. And so while the essence of while the exact details of what I'm interested in have changed and shifted, I think the essence of really what I was hoping to do is really still really central to my core self in 2006. And so I wanted to just end with a few notes to just highlight to you all that, from my life lesson, that having goals in life is a really important and amazing thing to do, but making sure that those goals don't inhibit you from actually pursuing other interesting opportunities and become limitations is also very important. And then I think that hopefully what I've also articulated to you all is that the discovery process, and we're all part of the discovery process because we're all doing research at UCLA, is an amazing process. And it's really what's one of the coolest aspects is it can lead to unexpected paths and turns. And so you never know where you're going to end up. And so with that, I want to thank you all for your time. I'm really excited to take any questions for anyone who has any. And I just thank you all for taking the time to listen. Thank you.
Thank you so much, Dewan. That was a wonderful presentation. I'm going to give you some
Thank
clapping,
you.
even though we can't see our audience. I can do my own clapping for you. And we have some really interesting questions, like literally covering sort of everything that you've talked about today.
Great, yeah.
So I'm going to start with some questions that were from individuals who typed in questions as they registered for the event. So the first question is, how did you actually decide which research to focus on and find out what you love?
Yeah, it's a good question. And hopefully you can see that I didn't really know. I just took some guesses and I just enjoyed the process. And what I found was that the most important thing that I chose was I wanted to be in a lab environment that was nurturing and supportive. And so as long as that environment was conducive to me being able to ask cool questions, then I was more than happy to say yes. And so even though I loved vasculature to this day, I think I made the right choices because I really love I love any kind of developmental biology question. But more importantly, I got the training and the support that I needed to carry me along the way to do something I really enjoy.
So the bottom line is, it isn't exactly what you study, it's the people that you do it with.
I think that's so important. It's underappreciated.
Yes. So we have some questions about just starting undergraduate research. So when would you suggest that our audience start undergraduate research? Should you start in the summer, or do you think starting in the school year is better? What is the best time, do you think? What year do you think they should start? Along those lines.
I started early, I gotta say, and I think starting early gives you the opportunity to be able to grow and watch a project really blossom. And so that was been like the best part was I stuck around for years and I got to develop a project and I got all these tools along the way. And so, I mean, I think the key is that what I think the most important thing is, when can you dedicate the time to actually do it? I think it's very hard to get the maximum experience when you only have 2 hours out of the week to contribute to science, because science is not it's not easy to do great fun science in very small periods. And so having like a full summer is an awesome way to actually get that experience going, because you have dedicated time to grow, and those skills will carry you throughout the academic year because they'll give you some confidence and you'll know how to do some of these skills throughout. But as if, there's a lot of labs here. So as soon as you can get in, I would say try to go for it. But I think you definitely want to make sure you have the time to allocate to that.
All right, everybody. I hope you heard that. Don't start research till you know you can say set aside 10 hours and you'll get an A in all your classes.
Exactly.
Right. Here's a question which just shows that not everybody knows how it works to get a PhD.
Yeah.
How much did your PhD program cost?
Yes, $0. Yeah.
$0.
So, PhDs, particularly in the biomedical sciences, are typically, funded. And so folks like me and the PIs, the investigators, the programs are funding you. And not only are they funding you, but you're getting paid throughout this process. And so, so it's one of those rare opportunities in the US, especially where, you can come out with no debt, but with a degree that can give you a job and a career.
Isn't that nice?
Yeah.
To come out with no debt.
Yeah, beautiful.
So here's a question about your motivation. So what do you think is your biggest motivation that keeps you going and continues your drive to work and study science and do research?
Yeah, it's a good question. And I feel like it changes over time, but a consistent theme for me and at a scientific level, what always interests me is how do cells interact with each other? How are these complex systems being able to communicate with each other and coordinate these processes? We're such complex beings, right? Just me moving my fingers is actually so many different systems working together, and they all have to coordinate and like do it in a real time. And so whenever I get any kind of question that I ask, or if you ever see me in a seminar, those are kind of questions that I'm going to ask about, because those are the types of interactions that drive my passion. But I am a developmental biologist. I didn't know it, and I think now it's very clear that I'm always interested in how things form and how they get more complex over time, and how that becomes dysfunctional or changes in the context of disease. And so anything within that spectrum, I would be happy to. It doesn't matter, liver, placenta, the skull, I don't care. It's all fun.
I agree.
Yeah.
I agree. So we have a few questions about engagement. And in particular, there was one student who was interested in your opinion on how we can effectively engage more underrepresented students in the sciences, in particular, kids from Compton.
Yeah.
What do you think about what we can do in that area?
Yeah. First, I think there's some good movements happening, but I think as a kid growing up in Compton, I didn't know that being a scientist was a career. I wasn't exposed to that opportunity. And typically, often it's the folks who have parents who have PhDs so now I'm in this privileged setting. So my daughter will know pretty clearly what a scientist looks like or what. And so I think that, and so trying to find ways to actually feed back and go back into the environment and actually demonstrate to them early on that this is something you can do. Because I think what happens at these early stages is people who are interested in science automatically put on the identity of I want to be a physician, right? Because that's the most clear marker for someone who has no exposure to science, because everyone sees a doctor. And so I think that the ability to get that exposure and bring people to UCLA is so valuable. And there's programs here that do that, that exist. But I think particularly like trying to find a pipeline for Compton schools to go into middle schools and elementary schools. I think that's the starting point, where just by showing them what it looks like at an early stage, it allows them to be able to perceive and see that it's possible. And then there's a whole bunch of things like the pipeline, making sure the pipeline isn't broken and making sure you're supporting these folks at each stage is also important. And I think at UCLA, I can see the growth in the years I've been gone that have been bridging that pipeline for once someone says they want to be a scientist, how do you make sure they wanted to be a scientist?
So we actually have a high school student in the audience who asked specifically, if you're a high school student, how do you find a research opportunity?
Yeah, and so that might be the hardest time, right? It's like, I have been in every lab that I've been in has had high school students do science, and so there are some programs out there, but a lot of it is just your self-initiative and saying, taking a risk and emailing someone and accepting that you're going to get a lot of rejections. As for undergrads too, there's going to be a lot of people who just don't have space or have time, but just taking that risk and saying, hey, I know I'm interested in the heart right now. And I see you do heart research. I'm motivated. I'm talented. I'm inexpensive. I'll volunteer. Can I be in your space? Other folks who are more privileged have programs at their schools that can help facilitate this process. And so if you don't have that, I think you just have to take that risk and say, I want to volunteer And it will take some time because scientists are busy, they're professors, they teach, they have to balance all these things. And so don't let rejection slow you down. Just be persistent and keep trying.
So what I do want to tell the audience is that as long as you haven't graduated from college, you're considered an undergraduate. So that means high school students get to do undergraduate research.
Yeah, nice.
We have another high school student who wanted to know if they could do research after high school graduation, but before starting college. And the answer is yes.
Yes, the answer is definitely yes.
But you just need to get in the door, as Dewan has
Yeah.
said. Okay. So what is the one thing you wish you knew before you started research as an undergrad?
Huh. And that's an interesting question. I don't know. Let me give me a second to this process. One thing I wish I knew. You know, I think for me, things just fell into place. But I think you know, like I enjoyed my experience working with Vicky Mays, right? But that was a fortuitous introduction that would not have happened, right? If otherwise, you know, who knows where my path would have led if I hadn't had that introduction. So I definitely think this goes back to, you know, another question about having exposure at an early level, because again, and even working with Vicky, it took me a long time to realize what it looked like to be a part of science in academia or in industry. And so having that knowledge base would have been really helpful. But for me personally, I guess it worked out so well that I can't say I will recreate it somehow. But I can imagine there's lots of other folks who don't have that exposure who would be making different decisions if they had known. I just got lucky and stumbled into it, but you know.
It helped that you were fun to talk to even back then.
Yeah.
So,
Oh, good.
so
Yeah.
I would offer that being outgoing and willing to talk
Yeah.
to me is a
Yeah.
huge step forward.
Yeah, there's so many opportunities at UCLA. There's so many ways to support you to doing research, to help protect your time. That, you know, I was very much about. You know, I would, you know, I was very nicely supported. You know, like Tama knew who I was from the moment she possibly could, and so and I stayed in touch, right, for years to come, right. Every year I was interacting, and you know, even after I graduated, I was still coming back, looking for ways that I can stay in science. And so I think you have to have that willpower to maneuver through to make it happen.
So you had mentioned earlier how you know, another option is going in industry, and there's a question from the audience asking if you think you want to stay in the academia and be a professor, or would you actually branch into industry?
Yeah, and so this is something that I have thought about, because I didn't want to choose a career in academia as a default, you know. And I think for me, the reason why I'm in this space is because there's many different things about being a professor that I enjoy. Obviously, I enjoy the science. I love the freedom to like change and do kind of any kind of science at will, assuming I have money. You know, that's the limitation there. But I also really love interacting with undergraduates, and I think, you know, I love to be in a classroom setting, and so these are the complements. These are the things that complement each other that make me really want to pursue a professor position versus you know, just a pure science position. Because when I was able to do pure science as a graduate student and a postdoc, I found that I was actually extending myself into these other mechanisms, because I wanted to be in touch with undergrad research. I wanted to be a mentor. And you can do some of these things in industry as well, but you can absolutely bridge these things. It's an expectation to do these things as a professor. And so for me, that was it. But there's plenty of people who are like, I just want to do the science, and the science is the most thing. And I love doing the experiments. And, you know, and then for those folks, you know, industry is a remarkable place. And it's such so much great science going on there as well, that there's no reason to have to choose between if you want to go into industry. You can do great science there with great funding. And so, yeah. So,
We'll work on that.
yeah.
So I have a science question for you, actually.
Yeah.
So I know that you have moved into this field of studying the cranium,
Yeah.
and how the skull develops. And I'm curious to know what you sort of shared with us your big aha moment previously when you did a knockout and found it affect the lacrimal glands when you were a
Yeah.
grad student. What was the aha moment that made you realize, oh, I really want to study the development of this unusual skeletal system that,
Yeah.
no one else has studied it. So that's good. It's nice to have your own specialty that
Yeah.
no one else does. But what was the aha moment that made you realize that is really what I want to study?
I think one of the most remarkable parts to me about this, the reason why I just love it in part, I can see it the entire time. How rare is it to actually be able to see the same part of the body? It's not going inside. It's not like, it's not being, like the heart, is great for a while and then it gets harder to see. But this is a process I can watch the entire time. And so it was just that, like, so the number of developmental biology questions that I can ask, because I can see it and I can manipulate it in real time, is something that was like, I don't know how to not keep this system, And then, there's obviously also like, there's such clear relations to like human disease that, it's like, even though it's a zebrafish, the exact same fusions happen in mice and humans and fish, it's something that's super remarkable. But I think I really love the imaging, like, because to me, like, to visualize a process as a visual learner is like such a powerful thing. And when you see something, it's very hard to argue against it. And so I think that was like the like, it's even studying the skeleton in other places, right? You don't have that advantage because they move in, right? They're surrounded by muscles, but the calvaria is just there waiting, And so I love it. I really do.
So a follow-up to that is, are you interested in traumatic brain injury? Because there's a lot of groups on this campus that actually are really interested in the questions of recovery from traumatic brain injury. They mostly have focused on how the brain itself recovers and how the immune system is engaged.
Oh
But
yeah.
I'm curious if you're interested in studying that as well.
Absolutely. And that's definitely, so right now, a lot of my research questions are focused on development, but I think what I'm hoping to bridge into is regeneration, right? And so how does the calvaria, like it needs to be repaired too, right? You have a traumatic fracture, like it has brain damage, but there's all these layers between the brain. There's another population called the meninges that wraps around the brain. All these structures need to be regenerated to actually heal. And there's definitely circumstances where this process does not actually go and you have to replace it with the metal component. So there's a need to understand this as well. So absolutely, and the great part is that zebrafish are one of the greatest models for regeneration. And so we can really begin to understand, what does it take for something to go right? In a case of like a zebrafish, and then we can go to a less regenerative model like the mouse and ask, okay, what's not going as well? And how can we actually stimulate the environment to make it go better? And so I think this dual system is going to be a really powerful model for that regeneration type of question.
So we have some questions about how you get money.
Yeah.
Just because they've never had anybody to ask before.
Yeah.
So this particular attendee is curious to know how hard it is to get funding to study what you do. And what happens if you don't get money?
I hope to never know. But let's say that. Yeah. And so as a professor, and so first of all, there's money as a trainee. There's fellowships out there as an undergraduate, as a high school student. There's money in the world to help fund you to do research. Once you get to this stage, it's really driven by the science. And so I, in large part, am funded through either private funding or through the most common mechanism, which is the NIH. And so there's actual independent institutes that fund different types of research. And there's actually an institute that funds craniofacial biology. And so what we do, and I think what every professor that has a research lab is doing at least a third of the time, right, is either writing or trying to think about what to write to actually show a new project and to convince a group of other scientists that this science will advance the human condition. And that's how you get funded each year. You have to demonstrate that you can actually do good science. and then you have to communicate the science that you want to pursue, and then you hope that the committees that have the power to make the decisions will be willing to give you that money. And so this is a cycle that every science professor is doing on a constant level, because it's this money that's going to fund the animals to be able to do the experiments, and the people to be able to actually do the experiments as well. And so it's grants and grants and grants, and it's all about writing, but it can be very difficult, right? Because there's a lot of people who want money, but you just have to be persistent. And it's a pretty, when I joined science, what I had to realize is that I am resilient enough to be rejected and keep going enough for this job. Because being a professor, being a scientist will require you to face rejection at some stage, no matter how talented you are. No one's going to agree with every idea that you have. And so resiliency is the key. I hope I answered that question well enough.
That was an awesome answer. And I think I know you're resilient, and I know all of our Bruins are resilient as well.
Yeah,
so
exactly.
we all can bounce back from all the challenges. So we're going to ask one last question, which is, looking back, what do you remember about the Undergraduate Research Center? And maybe you can tell the audience some of the things that you think that you would recommend for them to do.
Yeah, and so so I was there a lot. And so so after joining Louisa's lab, I was a part of SURF, and I got to be in the dorms, and like that's a really great one in part because, you know, as an undergraduate, you have an opportunity to go somewhere else and do research, or you can stay at UCLA and do research. And I was an assistant, I was a resident assistant, and so I had always had a need to be at UCLA at some point. And so I stayed, but I stayed in the dorms with all these other people who were coming from across the country to do science, right? And so I had really, really great memories of like meeting people who I know now who are professors at Berkeley and other places who are still doing science, right? But I had, you know, and we just had such a blast, you know, doing science, but actually coming at the end of the day and coming out and sharing our science experiences. And so that was something that was really fun for me. And then one of the great things that really helped me understand that I could enjoy teaching and like mentorship was there's a program called Bicep that I participated in but I also TA'd for. And that TA-ship did a lot for me because I really enjoyed it. And like, I found that I was like trying to stay as long as possible to engage with the undergraduates and help them show them and like be supportive to that process. And so that was a really great learning experience because it was my first experience with interacting with undergraduates and saying, you know, I can do this, and I enjoy this one-on-one interaction and showing someone how to do a scientific experiment. And so these are all through, the center, and this is just two things, right? But I did Bicep, I did all these things, and I was a MARC program, and so it was invaluable for me. And the funding and the support is something that I personally I needed that to be able to dedicate the time to research. Without that, I would have had to, spend more time working, and so it really did make it possible for me to really dedicate as many hours as I did to science, that turned out to be a lot. So I had a blast.
Well, we thank you, Dewan. We thank you for being a wonderful product of the kind of programs that we run. But more importantly, I really want to thank you for your engagement with all of us, for coming and speaking to all the students here as part of Research Week, for being so willing to take your time to engage with undergraduates. I know that you are going to be swarmed with MCDB majors who want to work for you and with you. And maybe we'll have you come see this year's Bicep group.
That would be fantastic. Please invite me. I'm here. Yeah. And so
I thought you started July 1.
So I started March 1st. I did a little off-cycle hire. And so so I'm here, and things are still slow, but I am here. And so thank you so much for inviting me. I really enjoyed preparing this and sharing as well.
Well, you are our final speaker as a showcase speaker for Undergraduate Research Week. For those of you in the audience, we are hosting at 2:00 our Undergraduate Research Week awards ceremony, where we'll be giving out medals to our Dean's Prize winners, as well as to the winners of our Faculty Mentorship Awards. as well as the winner of the library prize. So if you go back to the Research Week website and click on the events, you'll be able to see a link that will take you to the awards ceremony at 2.
Exciting.
And with that, I am going to close out this session. Thank you, everybody, for coming, and have a wonderful Research Week.
Thank you.